into therapies and benefits for the patients that we serve. So when I told you that the stem cell research here is as diverse as biomedical needs, here you can see an example. Tim Camp, one of the most prominent cardiologists and co-director of the UW Regenerative Medicine Center. If you look, Jamie Thompson, of course, the founder of the field, but if you look at the different applications, in the neurosciences alone, every single disease area, every specific disease, Alzheimer has its own laboratory. Parkinson's disease has its own laboratory. Multiple sclerosis has its own laboratory. In our case, we study the neurodevelopmental disorders. We have people looking at cancer. We have chemists and engineers. We have one of the most prominent bioethicists in the world, Altacharo. So not only do we perform the research, but we're also able to study how to take that to a commercial scale, for example, with the biomanufacturing facility here, which is led by Derek. Sorry. We don't need that on Judy's face. So this is just a snapshot. Not all the laboratories and scientists are represented here. We have more than 25 laboratories working exclusively on stem cell research on this campus. So let's start with the basics. What is a stem cell? What's the biological property of a stem cell? A stem cell, to be classified as such, needs to fulfill two requirements. First, it needs to be able to replicate itself, while at the same time, it needs to be able to differentiate into another cell, for example, a neuron. So those are the two exclusive properties of a stem cell. So we have different types of stem cells, as you've heard. The first type that you hear mostly about are embryonic stem cells. Embryonic stem cells, as the name says, are derived from human embryos. At day 5 after fertilization, there's this part, as you can see, in this stage, there's no differentiation. When I use this differentiation, I mean other tissues such as heart cells, brain cells, bone cells, fat cells, muscle cells, etc. At this stage, there is no differentiation. The embryo is composed of this inner cell mass. There's only two layers, the inner cell mass and this other layer called the trophectoderm, and this is a blastocele. It's a cavity filled with fluid. So from the inner cell mass, we excise the inner cell mass, and then we grow these cells in the laboratory. This is a colony of human embryonic stem cells. So this is what a colony of undifferentiated human embryonic stem cells look like. So these cells can be cued into becoming brain or heart or bone, etc. The other type of stem cells are adult stem cells. For example, inside our bone marrow, we have stem cells, adult stem cells. These can also be grown. This is a picture of bone marrow stem cells or hematopoietic stem cells. So what are the major differences between embryonic stem cells and adult stem cells? Embryonic stem cells differentiate into all the therapeutic cell types in the body. Adult stem cells do not differentiate into all therapeutic cell types in the body. The embryonic stem cells, they proliferate almost indefinitely. So it's been 10 years after Dr. Thompson, he discovered the landmark paper was in 1998. So, to give you a practical example, I am a human embryonic stem cell scientist. I have never touched a human embryo. Why? Because once a human embryonic stem cell line is derived, it grows indefinitely. Today, in year 2008, I'm still working on the cells that Dr. Thompson isolated in 1998. So if that doesn't tell you that these cells proliferate almost indefinitely, I don't know what other example I can give you. So they do proliferate almost indefinitely to create massive numbers of therapeutic cells to transplant into patients. Adult stem cells do not proliferate indefinitely. I've worked with these cells. You can have about 10, 20 passages in the cell culture dish, and then they senesce or they die. Human embryonic stem cells have not been tested in human patients. Adult stem cells have been transplanted very successfully into human patients. So treatments for leukemia, for example, that have been in the clinic since the 80s, that's an adult stem cell therapy example. If you want to know how many clinical trials are going on in the US today, you go to www.clinicaltrials.gov and you put the keyword stem cell, and you're going to see hundreds of clinical trials that are already ongoing, most of them with adult stem cells. So where do these come from? These come from surplus in vitro fertilization embryos with donor consent. So here's the story. There is the impression we have been associated, stem cell research has been associated with the fact and the misperception, it's actually not a fact, it's a misperception that human embryonic stem cell research creates the needs for human embryos. That's not true. Since in vitro fertilization clinics, and in vitro fertilization has helped so many patients, embryos have been discarded. People have to make a decision. If you're a couple and you go to an IVF clinic and the doctor produces 7 embryos for you, and he cannot transplant those 7 embryos into, he transplants 2 or 3. You have 4 left in the freezer. Either you choose to keep those frozen indefinitely, or you make a decision to discard those embryos. So that is where the embryos that we work come from. We're not creating a greater need. These embryos, once they reach the research laboratory, they had 2 paths. At that point, the decision's already been made. So you see how we, the scientists, have been blamed, or there is this misperception. At the point when the embryo reaches the research laboratories, the decision had already been made. Those embryos would be discarded anyway. So that's where human embryonic stem cells come from. With the donor consent, of course. Adult stem cells can be found in many different tissues, such as bone marrow, fat, and heart. Fat is one of the richest tissues with adult stem cells. So there are many groups studying liposuction aspirates. You take off the whole liposuction aspirate, and then there are certain markers in cells within the fat tissue. CD34 is one of them that confers them a stem cell property. Again, those 2 stem cells, to be a stem cell, you have to self-renew, create copies of yourself, or differentiate into other tissue. So the promise of regenerative medicine relies on the fact that human embryonic stem cells derived from the inner cell mass are pluripotent, meaning they can differentiate into every cell type of the body and make cells such as liver cells, heart cells, bone marrow cells, and brain cells. And we also have the adult stem cells that I talked about. Remember when I said they do not differentiate into all cell types of the body? They're multipotent, they're not pluripotent. Does that mean one research avenue is better than the other? Absolutely not. So when I'm posed, often I get this question, well, why do we have to work with human embryonic stem cells? Can't adult stem cells fulfill the promise and the opportunities in regenerative medicine? The answer is no. Today, no. It would be the same thing as if 50 years ago, 60 years ago, when we were trying to develop therapies for cancer, somebody had asked you, okay, you could just study chemotherapy or radiation therapy. You can't study both. And now we know that depending on the cancer, radiation therapy is recommended, or chemotherapy, or both. So I see in ourselves in developing the next generation of regenerative medicine that we are posed in the same context. So the benefits of stem cell research can be divided in 3 categories. The first is regenerative medicine and tissue engineering. The second is the applications of stem cells in drug discovery, toxicology, to better understand adverse reactions. And the third is, strictly speaking, biomedical research, where we understand the mechanisms of disease and human development. For